Hello everyone, welcome back to Biology Board. This is BRIJESCHOTALIYA and today we will going to study about DNA. So we will easily study about DNA, its replication, its genetic code in an easy way and in very short. Okay, so let's start with the DNA. Everyone must be familiar with the word DNA, right? What is DNA? DNA is a deoxyribonucleic acid. Yes, DNA stands for deoxyribonucleic acid. What is DNA? This is a chemical substance which is present in nucleus of all cells in all living organisms. Everyone must be knowing that living organisms are made up of cells and cells contain nucleus and nucleus contains the genetic material which we call it as DNA. DNA controls all the chemical changes which takes place in cells. Okay, DNA controls all the chemical changes which takes place in the cell. The kind of cell which is formed, that is muscle cell, blood cell, nerve cell, any sort of cell, it's controlled only by DNA, that is deoxyribonucleic acid, and it's our genetic material. The kind of organisms which is produced is controlled by DNA. Any kind of organisms, they must contain DNA. Without DNA, there is nothing possible. Every cell contains a nucleus and nucleus contains genetic material. DNA is a very large molecule made up of a long chain of subunits. DNA is a large molecule which is, let's say this is DNA. It's a large molecule. It contains many type of subunits. Like this one, it's a very large molecule. Okay. The subunits are called nucleotides, and this is known as nucleotides. Each nucleotide is made up of a sugar called deoxyribose, a phosphate group is present, and an organic base. Okay, there are three substances required for a nucleotide. Ribose is a sugar like glucose but with only 5 carbon atoms in its molecule. Ribose is a sugar like glucose, but glucose is 6 carbon atom molecule, but ribose is a 5 carbon atom molecule. Deoxyribose is almost the same but lacks 1 oxygen atom. Hence, DNA is the most stable genetic material. RNA is not a stable genetic material, but DNA is the most stable material since it lacks 1 oxygen atom in its molecule. Both molecules may be represented by this symbol. This is 1, 2, 3, 4 and 5. 5 carbon atoms present in its molecule. Okay. The bases. The most common organic bases are adenine and thymine and cytosine and guanine. Okay, these are the 4 organic bases present in a DNA. The deoxyribose, the phosphate, and one of the bases. This combines and forms a nucleotide. This is only one nucleotide. 3 substances combines and one nucleotide is formed. Then nucleotides are joined and a molecule of DNA is formed by millions of nucleotides joined together into a long chain. How they are joined? A sugar phosphate backbone. This will be the backbone. Okay. This all will be the backbone and this will be the bases. The bases will get attached with another. Since we know that DNA is a two-stranded DNA molecule. Okay. So this is all about one strand and another strand will combine with the bases. In fact, the DNA is usually consists of a double strand of nucleotides, as I have told. Okay, the sugar phosphate chains are on the outside and the strands are held together by chemical bonds between the bases. Seen here, sugar phosphate bond, they are held outside and bases are at the inner side, held together by chemical bonds between the bases. Two-stranded DNA. This is the one strand as we have seen earlier. This will get attached by the specific type of organic bases, that is, see this animation. Bonding. The bases always pair up in the same way. Adenine forms a bond with thymine and like this one. Okay, blue one is adenine and let's say this is thymine. And cytosine always bonds with guanine. Cytosine bonds with guanine. Okay. These A and G are purine bases and T and C, they are pyrimidine bases. Okay, I have not mentioned this purine and pyrimidine bases, but understand by yourself. Bonding 2. Getting the point? Adenine always binds with thymine, cytosine always binds with guanine. Like same way, this is thymine and this is adenine. This is guanine and cytosine. This is the backbone. These two are the backbones. And bases always held together by the specific their bond. Adenine always binds with thymine, cytosine always binds with guanine, and these are the sugar phosphate backbones. So this had become a DNA molecule. Pairing up. How does pairing up takes place? See this animation. This is how they paired up. Okay. The paired strands are coiled into a spiral called a double helix. Okay. These gets become paired and now they are twisted. The DNA is a double helix molecule. The double helix molecule, this is how it's look like DNA. The DNA looks like this one. See, this is two strand, adenine bind with thymine and guanine bind with cytosine, and helix is formed. It's a look like a circular ladder. Okay, sugar phosphate chain. This is sugar phosphate chain going all through this. These are the bases. This is a DIY model of part of a DNA molecule. Before a cell divides, the DNA strands unwind and separate. We are talking about replication here. Before a cell divides, DNA strands unwind and separate. First, this was the coiled. Now first of all they unwind, they become a straight like this one. And they get separate by some enzymes. Each strand makes a new partner by adding the appropriate nucleotides. Let's say this is the one base, this is the one strand, this got separated. Now this contains a backbone of sugar and phosphate and one base, and this also sugar and phosphate and one base. Okay, now how does DNA replication takes place? There are some specific enzymes, we are not going in detail, but this pairs up when a specific nucleotide is added and a strand is formed like this one. Okay, hence we get two DNA molecules from a single one. This is how replication takes place. Let's say this was the adenine, then here comes a specific nucleotide which is thymine. If it's G, then comes C. Okay. The result is that there are now two double-stranded DNA molecules in the nucleus. Due to replication, we get two DNA molecules. So that when the cell divides, each nucleus contains identical DNA. Are you getting the point? When the cell divides, each nucleus contains two identical DNA as the same nucleotide pairs up with the another nucleotide. Okay. This process is called replication. The strands get separate like this way. Okay. Each strand builds up its partner by adding the appropriate nucleotides. As we have seen, this was one strand broken. Let's say this was A, this was T, this was G and C. A specific nucleotide will come and get bind with here. Let's say this was A, then the opposite will be T, then A, G, then C, then G. A always binds with T and G always binds with C. See this animation. So like this way, we get two DNA strands. Okay, understand with the term? If you have any doubt, you can comment in the comment section and let me know. Everyone will get this PPT. I will share the link in the description box. You can download from there. Okay, let's start with the genetic code. The sequence of bases in DNA forms the genetic code. DNA, this is DNA, sequence of bases, this is DNA, this forms the genetic code. Is a group of three bases, a triplet controls the production of a particular amino acid in the cytoplasm of the cell. So let's take a small cut of DNA. This is called triplet code. Let's say this is A, C and G, A, T, C, then G, G, C. These three nucleotides combines and it forms a triplet. Triplet is a type of gene which controls the activity of the DNA. How it controls? The triplet controls the production of a particular amino acids. These three is responsible for production of particular amino acids. The different amino acids and the order in which they are joined up determines the sort of protein being produced. These amino acids produce a protein. Here you will get the point. This is a small imaginary protein molecule showing how a sequence of 5 different amino acids could determine the shape and identity of a molecule. This is it. These are the amino acids. These all are the amino acids. Okay. Each amino acid, serine, cysteine, valine, glycine and alanine, is coded for a particular triplet of bases. What is triplet? Sequence of 3 nucleotides is known as triplet. For example, cysteine, adenine, cytosine, adenine and thymine. This codes for a valine. These three, that is CAT. This codes for one specific amino acid, that is valine. In the same way, cytosine, guanine and adenine, CGA, this codes for a specific amino acid, that is alanine. Like in same way, all nucleotides in the DNA, they forms a triplet, and these three triplets codes for one amino acid. This is known as the triplet code. Familiar with the word now? What is triplet? Okay. Each triplet codes for a specific amino acid. Each triplet code like this way. All codes for a specific amino acid, like CGA codes for alanine, valine, glycine. This is glycine, arginine. like this way. Okay, each triplet codes for a specific amino acid. The amino acids are joined together in the correct sequence to make a part of a protein. Okay, this protein is a long chain of polypeptide of amino acids. They are polypeptide chains. These all get bind with a peptide bond and a protein is formed. Like this one, DNA. This is a protein. All amino acids get bind with peptide bond. Okay. First the chain was called as polynucleotide chains when they were not converted into amino acid chain. A triplet was not formed and amino acid was not converted. Then after conversion of amino acids to make a protein, they are binded with peptide bonds and a polypeptide chain is formed. Then DNA and enzymes. The protein builds the cell structures. They also make enzymes. Look, there's a term. All proteins are enzymes, but all enzymes are not protein. Okay? This is the main point you all should know. The DNA controls which enzymes are made and the enzymes determine what reaction takes place. The structures and reactions in the cell determine what sort of a cell it is and what is its function. So DNA exerts its control through the enzymes. Hope you will be familiar with these words. Genes. A sequence of triplets in the DNA molecule may code for a complete protein. Such a sequence forms a gene. Okay? There may be 1,000 or more bases in one gene. What are bases? Let's say this is DNA strand. There may be 1,000 or more, and this will call it as gene, and the sequence will be known as a gene. So there are questions for you. Which of the following are components of nucleotide? You must answer and do it yourself. I will be giving you the answers in the description box. These are some questions. Then, which of the following represent the correct pairing of bases? DNA molecules are formed from which of the following are organic bases? Replication of DNA occurs. A nucleotide triplet codes for. If you download the PPT, okay, and if you touch this green button, you will get the correct answer. If it will be a right, then after touching, it will give you the answer. The option which I touched, that was incorrect. So nucleotide triplet codes for an amino acid. So if I touch this B option, the answer is correct. If I touch for an enzyme, the answer is incorrect. If I touch an organic base, the answer is also incorrect. So the answer was an amino acid. Hope you will solve these 6 questions and will like this video. If you have any doubt, you can comment me in the comment section. Thank you for watching this.